Again, I would like to welcome everyone. My name is Jakub Lewicki. I'm a business development manager here at Pelago Bioscience. And welcome to our third webinar in the series of webinars that we organized this year with some of the partners and clients. So probably most of you already know Pelago as a company, but for those who are new in the our new friends, we are a specialized CRO working with CETSA technology, and again we are based here in Stockholm in Sweden, and we will talk more about CETSA technology today. So the series of events was created to introduce our audience to CETSA technology, its applicability across drug discovery pipeline. And so far in the series of events, we covered a target ID with Mary Sue from Pfizer. And we also had the previous webinar about lead generation by Dr. Joseph Shaw from AstraZeneca. So both of these webinars are actually available and recorded to watch on demand. You can find the links on our LinkedIn page if you're interested. And today we are moving actually a step forward in the process of drug discovery. Today on the webinar, we will focus on preclinical studies. So before we start, I would like to introduce our today's speakers. So first, we will have Dr. Peter Joyce, CEO of Grey Wolf Therapeutics. He will introduce you to the company as well as the project in general. So Peter is founder and chief executive officer of Grey Wolf Therapeutics, setting up the company in 2017 together with Tom McCarty. Peter has now over 15 years of experience in biomedical research, leading drug discovery projects across disease areas. So prior to Grey Wolf, he was at Vertex Pharmaceutical for 5.5 years, where he led drug discovery projects and had responsibility for leading a number of target ID teams. And before Vertex times, he was a postdoc at the MRC. Peter holds a PhD in biochemistry and molecular genetics from the University of Bristol and a Bachelor of Science in biochemistry. And together with Peter today, our next speaker will be Dr. Juliet Morgan from Sygnature Discovery. She will walk you through the details of the experiments and results from this screen. So Juliet has Master of Chemistry degree from University of York in biological and medicinal chemistry. She completed PhD at the University of Nottingham in 2016, focusing on biophysical techniques to probe protein-protein interactions, and in particular applications of SPR and high-field NMR methods. Now, Juliet is a senior scientist in bioscience department at Sygnature Discovery, specializing in biophysical and biochemical assay development. She's currently leading the Sygnature bioscience teamwork, focusing on the Grey Wolf project. I would like to thank you again for participating, both the speakers and all the attendees of the webinar. I would like to encourage our audience to use chat function on Zoom. So I would ask you to have your microphones muted, but if you would like to ask any questions throughout the presentation, please type them in the chat box so we can have later at the end of the webinar QA session. So yes, without further ado, Peter, stage is yours, the virtual stage. So please, you can start with your presentation.
Great, thank you. Juliet, you're right to share the slides from your side.
Yeah.
Thanks for the kind introduction and for inviting us both to come and talk you through the projects that we've been doing at Grey Wolf and how we've used the CETSA platform really as a key part of our discovery engine. So yeah, just by means of an intro, so Grey Wolf Therapeutics, we're a small molecule drug discovery company working in immuno-oncology. And the premise behind the company is that we're trying to generate these non-self peptides, neoantigens, in the context of cancer, really with the purpose of making these tumor cells more visible to the immune system, so that the immune system can detect the cancer as foreign and then ultimately lead to their destruction and cures in patients. And so if you move to the next slide, Juliet. So on the left-hand side, just I thought I'd give you a bit of context behind the company and then the purpose behind our programs before diving into the detail of CETSA, and I'll hand over to Juliet to take you through some of the really nice assay development work that's gone on here. So on the left-hand side here, this is data from non-small cell lung cancer treated with revolutionary drug called anti-PD-1 or Keytruda in this context, or pembrolizumab by its generic name. And what you're seeing there is actually a remarkable effect. Over 5 years, you're seeing a number of patients actually showing very durable benefit. But what you can see in the green line is there's still an awful lot of room for improvement. And it's a general consensus in the field that in order to drive further improvement, you need to combine these types of molecules, which essentially take a break off the immune system and lead to further activation of the immune system. You need to combine these checkpoint inhibitors with truly orthogonal approaches. And that's where we think these enzymes, ERAP1 and ERAP2, come into play. And interestingly, when you look in the cancer genome atlas and you look at the level of expression of this target ERAP1 in the tumor, you see that actually low expression of ERAP1 in the tumor correlates with quite a significant improvement in overall survival. So therein lies our kind of therapeutic hypothesis based on human genetic data, showing that if we were able to block this target, we could lead to a dramatic effect on overall cancer outcomes. So if you jump to the next one, Juliet. And so what is the target and what is it we're aiming to do? So the enzymes ERAP1 and ERAP2 reside in the endoplasmic reticulum of your cells, and they have a role in this pathway called the antigen presentation pathway, which is depicted here. You can just build the slide, Juliet, actually. And so proteins on the left-hand side are continually turned over by your proteasome into peptides. These peptides make their way into the R through that purple complex, which is called TAP. And at this point, the majority of these peptides will get loaded onto the orange complex called MHC class 1, shuttled to the surface to be seen as self or non-self by your human immune system. Now, what's interesting is the ERAP enzymes. These are aminopeptidases, so soluble proteins within the ER that trim peptides from their N terminus. And they're affecting a subset of antigens, a subset of peptides that make their way through this pathway. What's interesting is when you block the activity of this enzyme, you can drive two types of changes on the surface of a cancer cell. And the first change actually relates to the fact that you can drive entirely novel neoantigens. So these novel peptides come about probably through things like mutations within cancer cell DNA and can in fact be detected as foreign by the immune system. And then the second type of change, you can actually lead to some upregulation of these neoantigens. So you can actually dial up a pre-existing immune response. And it's really those two types of changes that drive the visibility of a tumor and can lead to these really quite powerful anti-tumorigenic T cell responses that before did not exist without an ERAP1 or an ERAP2 inhibitor. So if you jump on to the next slide, Juliet. Yeah. And so neoantigens, so these are, as I said, these are come about by often by mutations in cancer DNA that get transcribed and translated into peptides with these point mutations and can in fact be seen as non-self, and so call out the cancer cells as being different. And they're fundamental to an anti-tumor response. Without these, you will not get recognition of tumor as different and ultimately a destruction by cytotoxic T cells. What's also really interesting is that the greater number of these neoantigens in cancer, the better the response. And we use a correlate for neoantigen, the amount of neoantigens. The correlate we use for that is tumor mutation burden, so the number of mutations per megabase of DNA, and that's what this graph is showing you. And what's really quite striking is those cancer cells, those cancer types that have the greatest number of tumor mutations, so things like skin cancer caused by UV irradiation or non-small cell lung cancer often caused by smoking, they actually respond the best to checkpoint inhibitors. And that is because when you release this brake on the immune system through this drug anti-PD-1, you activate the immune system, and then they're more likely to see these cancers as foreign. Now, what we're aiming to do with our Grey Wolf inhibitors, our ERAP1 inhibitors here, is to try and drive even better responses in those patients that are responding and have high neoantigen burden, but also increase the overall response rate. So if you jump onto the next one, Juliet. And so that therapeutic hypothesis is really distilled here. So through the work that Juliet will walk you through, we've spent a lot of time really understanding the mechanism by which our molecules bind and building confidence in the ability of these molecules to not only work enzymatically and in biophysics assays, but importantly as we move into cell systems. And that's where the CETSA system has really come into play, is to build that confidence in that relationship between our enzyme data and true target engagement in cellular systems. And then it's the question of how does that data then correlate with changes in the neoantigen repertoire? Because what you could see on that previous diagram is there's a bit of a gap, a bit of a difference in the signaling pathway between the point at which we inhibit ERAP1 and then the change that we'll see on the surface. And that's really where CETSA's come into play, is the ability to understand target engagement in that cellular environment. So ultimately, inhibition drives neoantigen generation, as I've talked about, then leading to a differentiated T cell response. We activate an entirely novel T cell response that ultimately leads to tumor growth inhibition. But the focus of today will be focusing on the work that we've done to understand the landscape of our inhibition of the ERAP1 enzyme in that CETSA assay. So I think at this point, I'll hand over to you, Juliet, to take you through the complexities of ERAP1 isotypes.
Great. Thanks, Pete, and good afternoon. So I'm going to start this section of the talk by taking a close look at ERAP1 itself. So this is shown top right here, and it consists of 4 globular domains. And if you look at the domain schematic below, you can see the active site is located within domain 2, and the peptide binding site is actually formed from a cavity at the interface between domains 2 and 4. One of the key challenges we had when working with ERAP1 as a target is it experiences a high degree of polymorphism. And in fact, a total of 10 distinct haplotypes, and these are collections of characteristic multiple SNPs or singular nucleotide polymorphisms, are required to describe 99% of the population. And since the precise structure of the active site and the peptide binding site confer specificity and substrate handling, an individual SNP is capable of influencing enzymatic activity. And from this, in combination, this leads to significant variation across the ERAP1 haplotypes. So I've summarized the haplotypes here in this table, and they're notated HAP1 to HAP10 as they incur an increasing number of SNPs. And this makes HAP1 and HAP10 the 2 most divergent variants. It's useful to note that HAP2 is the most abundant form of ERAP1 in the population. Now, as I said, the SNPs result in significant variation across the haplotypes. And this is because they're occurring at key locations within the sequence. And we can look at this in a bit more detail. So residues 346 and 349 are found proximal to the catalytic site, while residues 528 and 575 are actually found within the region that undergoes conformational rearrangement, moving from the open to the closed form that confers increased enzymatic activity. And finally, residues 725 and 730 are located within the peptide binding site. So you can quickly see how in combination it gets quite difficult to predict enzymatic activity, and we're looking at quite a varied background across which to understand the ERAP1 behaviour. And this was what led us really into choosing CETSA to support the ERAP1 inhibitor programme. So as Pete highlighted, the end goal of ERAP inhibition is the generation of novel neoantigens. As you can imagine, the functional cellular assays required to observe this are complicated both in execution and readout. However, what CETSA provided for us was a method by which to directly observe our inhibitors engaging with the target in relevant cellular systems using a few relatively straightforward steps. Not only were we able to demonstrate target engagement, but we were also able to use it to generate EC50 data to understand compound potency and compound SAR. And further to this, as CETSA is agnostic to both target and substrate, it meant we could use one methodology to look at all of our ERAP1 inhibitors. And all of this was working towards building our understanding of how the ERAP1 polymorphism would affect the efficacy of our inhibitors in a patient population. So it was really important for our aims to make sure we were working in the right cellular systems. So we selected a range of cell lines which endogenously expressed ERAP1 and represented a range of the haplotypes in order to make sure we were covering the full spectrum of ERAP1 functionality. So we have examples here of HAP1 and HAP10. So these, as I said before, are the most divergent forms of ERAP1. And then alongside this, we're also looking at heterozygous variant examples. So SKMAL5 cells and the P3C3 cells are actually variants that combine haplotypes from either end of the spectrum. So looking at a 1-8 combination and a 2-TUM combination. And finally, we also selected CT26 cells. This was our intended in vivo study model and expresses the mouse homolog MERAP. So the first step in setting up a CETSA assay is to establish a melting temperature for your target of interest with or without compound treatment. And the process is outlined here. I'm just going to briefly run through this for anyone not familiar with the Navigate methodology. So you start out with cells and you incubate these with a single concentration of your tool or reference compound. After treatment, these cells are then subjected to heat challenge across a temperature gradient. The cells are then lysed and the soluble and insoluble fractions are separated by centrifugation. The soluble fraction is then analysed by Western blot. So the premise being that if your compound binds the target, it will stable the target against heat denaturation. However, in the absence of stabilisation, as the temperature increases, your target will unfold, aggregate, and be pulled out of the soluble fraction and be lost in the insoluble fraction of the material during centrifugation. However, the stable compound stabilized target will persist in the soluble fraction to higher temperatures as it remains folded in the presence of the compound. And this is what you analyze here, looking at the proportion of your soluble target, your target present in the soluble fraction. And if you plot the respective soluble fractions for both the compound treated and untreated samples, you can generate the melt curves as shown here. And you can see target engagement is observed as an increase in the melting temperature at these 2 points. So you've got the unstabilized target melting and denaturing here, but in the presence of the compound, we've seen a slight right shift and an increase in the melting temperature. And this is your target bound to the compound is stabilized and your melting temperature increases. So for initial assay development, we chose to work with 2 cell lines. So the CHAR cells express the haplotype 2, which is the most abundant form of ERAP1, and also the CT26, which express the mouse homolog MERAP. So we incubated these cells at 37 degrees with one of our early hit compounds, or DMSO, for 1 hour. And after this time, the cells were subjected to heat challenge over a temperature gradient of 37 to 63 degrees. We then lysed the cells using 3 cycles of freeze thaw, and the soluble fraction from the generated lysates was analysed by Western blot. And that's what we've got at the top of this figure here. So you can see the example from the CHAR generated lysates, and we're showing temperature gradient for with compound or with DMSO, which was the vehicle in this instance. And what you can see is as the temperature increases, you get to a point where you start to see, in the presence of vehicle only, you're seeing your response to the ERAP antibody fade, but the compound treated ERAP is seen to persist to higher temperatures as it is stabilized. So both the generated MELT curves are shown below here, and in both instances, the proportion of soluble MERAP or ERAP1 response is normalized to the amount present in the 37-degree condition. So this is essentially before the heat challenge starts. So it's going to be your highest concentration before the compound target faces any sort of denaturation or destabilization. And what we can see in both instances is in the presence of compound, we see a right shift and an increase in the TM value obtained. So we were able to demonstrate that both the mouse ERAP and the ERAP1 haplotype 2 were stabilized in the presence of our early hit compound, confirming binding and validating target engagement. We found that the MERAP was slightly more thermostable than the ERAP1 haplotype 2 form, with a MELT temperature of the vehicle only for 57 versus 51 degrees. Interestingly, we saw a smaller increase in the MELT temperature for MERAP over the ERAP1 hap2 in the presence of the early hit compound. And this is an early indication of the potency for the compound for the two forms. So findings summarized here in the table on the right. And we used this, so the comparison of the vehicle only and the compound treated MELT temperatures to select a temperature that we could use for concentration response experiments. So this is just identifying a point at which we're going to see the biggest differential, so the biggest assay window between the responses for compound stabilized or no stabilization. And this sits just between your vehicle only and your compound stabilized values. And as a point of reference, this is essentially your EC80 value. So moving on now to look at the EC50 rankings of the compounds. So this follows roughly the same core steps as we used to generate the MELT curves. But in this instance, the cells are now incubated with compounds across a range of concentrations. And then they're subjected to heat challenge at just the single temperature that had been selected. And then as before, lysed and the soluble fraction separated by centrifugation. Now, in this instance, you're normalizing the amount of soluble form of your target against your top concentration treatment, as this is likely to be the point that experiences the greatest degree of stabilization for your target. If you plot that against concentration, you're able to generate curves from which you can determine your EC50 values. So we worked again with the CT26 and CHAR cells. We incubated these for 1 hour, 37, with a 7-point compound serial dilution, with the top concentration of 200 micromolar. After this treatment time, the cells were subjected to the heat challenge at their respective temperatures. So this was 58 degrees for the CHARs, for the CT26 cells, which was slightly more stable, and 53 degrees for the CHAR cells. They were then lysed as before by repeat cycles of freeze thaw, and the soluble lysate fraction collected and analysed by Western blot. So as you can see here, in each case, the amount of your target is detected, the anti-MERAP or ERAP1 response is detected in your soluble fraction for each concentration condition and normalized against the highest concentration condition. This is then plotted to determine EC50 values, and these are summarized in the table below. And as you can see, in line with what our previous or earlier findings showed, we're seeing that our early hit compound has a slightly lower potency against MERAP as opposed to ERAP1 haplotype 2 that's expressed in the CHAR cells. And the two values just show the results that were obtained when the assay was transferred in-house to Sygnature, and as you can see, these show good agreement and testament to the robustness of the assay setup. So to summarise, we were able to find EC50 values for an early hit compound against both MERAP in CD26 cells and ERAP1 haplotype 2 in the CHAR cells, and these replicated when transferred to Sygnature. We made some modifications at this point. So the original concentration dose response curve was 7 points, which was adequate for these micromolar affinities. However, if you look at the CHAR compound, which is coming out sort of around a micromolar, you can see we're just sort of cutting the bottom of that curve. And if we go much further below that, the assay sensitivity is really going to struggle to define EC50s lower than that. So we decided at this point to reduce our top concentration to 100 micromolar and increased our dose response to an 11-point curve. And this all just hopefully providing us with a more robust setup and just gives us the ability to detect a wider range of potencies as we go into compound development and screening. So at this point, having sort of had a successful initial assay development in the 2 cell lines, we wanted to expand the CETSA assay to look at additional cell lines and be able to provide data for the program across the range of the ERAP1 haplotypes. So just as we've done for the other 2 examples, we obtained a melt curve for each cell line with and without treatment from the early hit compound in order to identify suitable temperatures that we could then go forward and use for the concentration response experiments. So these melt curves are shown here, and the data outlining their haplotype type and their melt temperature for the vehicle only, and then in the presence of compound treatment is summarized there as well. I'd just like to draw your attention to the melt curve for the NCI H23 cells. So these express haplotype 2, ERAP1, and compare that to the HCT15 cells, which express ERAP1 but the haplotype 10. So nearly the 2 most divergent, sort of the 2 pretty much opposite ends of the ERAP1 functionality. As you can see, we are seeing here quite a big difference between the 2 and sort of reinforces why we needed to do this characterization. Interestingly though, if you look at the heterozygous examples, so the SKML5 and the PC10, which sort of represents a mix of those 2 extremes, actually you're seeing somewhere between both of them, but possibly closer really to the HAP2 profile. So what was our findings regarding sort of the haplotype spectrum here? So in actual fact, we found thermal stability of the ERAP1 haplotypes, HAP1 to HAP8, was actually relatively similar. So we're looking at just the vehicle only melting temperatures, pretty much all of them were 51 to 52 degrees. However, the HAP10 variant of ERAP1 was shown to be significantly less thermally stable, with the vehicle melting temperature for that construct being 45 degrees C. Now, because of this, this meant in order to obtain a viable assay window, we were going to have to conduct our concentration response experiments for the HCT15 cells at much lower, so at 47 degrees C. However, actually, because the remaining haplotypes had a relatively similar melt temperature, we were able to continue using 53 degrees, which we'd identified for the CHAR cells, to evaluate all of those cell lines. So just a point I wanted to pick up on here, when you're making direct comparison of compound potency, you just need to keep in mind the differences in energy into the system. If you use a lower temperature for the heat challenge, if you do a direct comparison with another cell line at a higher temperature, it's going to over-predict your compound potency because you're not really subjecting the target to the same level of stress, same level of denaturation. So just to illustrate this, shown on the right, we've got an example where we measured the EC50 in the HCT15 cells, which express the HAP10 ERAP1 variant at both 47 and 49 degrees. And what you can see is as the temperature increases, the target's subjected to greater challenge. Actually, the ability of the compound to stabilize it decreases. And you'll see a further right shift of the potency. And this just gives you, looking at the table, an indication of really what the EC50, what direction the EC50 values would really be going in when you relate them to the other cell lines which are subjected to heat challenge at 53 degrees. Now, as the program progressed and more compounds went through the CETSA assay, we were able to show generally good correlation between the CETSA generated EC50 potency values and the respective haplotype biochemical assay EC50 values. And this plot on the right here shows a comparison from the CHAR cells, which express haplotype 2, against biochemical assay which used recombinant HAP2 ERAP1 construct. Now, at the beginning of the program for early hit molecules, we were observing quite a significant drop in potency for our inhibitors when we were looking at a comparison of HAP2 to HAP10 engagement for the ERAP1. And CETSA really played a key part in providing the data that we needed to understand this and closing this potency gap in order to ensure, as our molecules progressed into lead optimization, we were really achieving the best coverage across the ERAP1 spectrum. So what did we learn about the ERAP1 haplotypes from this work? So from all of the ERAP1 haplotypes we evaluated, we found our compounds had the highest potency against the HAP1 and HAP2 ERAP1 variants, and lower potency was observed for the HAP10 ERAP1 variant. Interestingly, we weren't quite sure what we were expecting when we started out on this. For the heterozygous variant, so that's the SKML5 cells and the P3C cells, which sort of are a combination of the two extremes of the ERAP1 spectrum. Actually, what you seem to find is the HAP1 or HAP2 present in these combinations is having actually the greater influence on the observed profile. So actually it's sort of skewing them more to that end of the spectrum. So just quickly, I should have just said, the table on the right just shows a profile for one of our development compounds. So you can kind of see the variation you're picking up across the different cell lines and across the different haplotypes. So I just quickly want to introduce the second target. So the second target is ERAP2, and polymorphism is actually far more limited in ERAP2. There's only one really significant polymorph, and this is associated with the K392N mutation. However, as the N392 variant is actually strongly associated with another SNP that results in impaired protein expression, the lysine 392 variant dominates within the population. And in fact, 25% of the population fail to express any ERAP2 at all. So in this case, as ERAP2 is not actually expressed in all individuals, the first step of setting up this assay was to identify a suitable cell line in which we could characterize the ERAP2 target engagement. So we found the breast cancer cell line HS578T actually expressed quite high levels of endogenous ERAP2 and comparatively low levels of ERAP1. So it's just a comparison here on the right. It shows these cells against CHAR, which was one of our workhorse cell lines, the ERAP1 CETSA assay, and also the SKML28 cells and PC3 cells, which we also did some ERAP1 characterization in. And you can see that we're getting significantly higher levels of ERAP2 in just the HS5728 T cells, minimal amounts seen in the SKML28 cells, and actually none at all is present in the CHAR cells. And compared to the other three, the HS578 T cells have relatively low levels of ERAP1, which is preferable at this stage. So we chose to move forward with this cell line for the ERAP2 CETSA assay development. So exactly the same steps as before, a MELT curve was obtained for the HS578 T cells, and this time just with or without an ERAP2 active site inhibitor. And we use this to identify a suitable temperature for subsequent concentration response studies. And from this, looking at the table on the right, you can see ERAP2 slightly more thermally stable than our typical ERAP1 variant. So we're seeing a melt temperature for the target on its own of 54 versus sort of the 51 to 52 degrees we were typically seeing for most of the ERAP1 haplotypes. We were able to demonstrate ERAP2 stabilization in the presence of the proposed active site inhibitor, confirming binding and target engagement. You'll notice it is quite a minimal stabilization, so it's of a relatively low potency here. So we were able to confirm the target engagement for the ERAP2 assay for a proposed active site inhibitor, and this assay is now in place as part of the ERAP2 program going forwards. So just to summarize really and wrap up, I hope I've been able to give you a flavour of the benefit that CETSA has had for this program. I think it's key to keep in mind the value it's added in the data we were able to access through very straightforward assay, the understanding this has added to for how we built a picture around the ERAP1 haplotypes and the variation we were going to face there. And it prepared us much better for understanding how we could achieve the best possible efficacy across the haplotype family, to make sure in the patient population the compounds would have the broadest engagement. We were able to show direct target engagement, and this was in cellular systems to the benefit of not having to do at this early stage when we're sort of driving compound SAR, the complicated functional cellular assays that were required for sort of observing novel neoantigen presentation. We were able to look really in detail at the ERAP1 haplotypes and understand what a compound's engagement would look like against both a homozygous and a heterozygous variant background. So I don't think we really understood before we went in this what that might look like. And finally, we're going to apply the same methodology for the ERAP2 program. So while it doesn't exhibit as many polymorphs as ERAP1, I think CETSA is still going to play a key role in this program, again, just triaging the compounds prior to testing in those sort of complicated functional cellular assays. So I'd just like to acknowledge all the people that worked on this. So the initial assay development for the CT26 and CHAR cells was done by Jacob at Pelago, the Grey Wolf team, Pete, Martin and Andrew, and finally my colleagues at Sygnature, who've all contributed to the work in the CETSA assay. We're happy to take any questions.
All right. Thank you very much for a presentation, both of you, Pete and Juliet. So really nice data, really great presentation. Thank you again. And I think that's a great moment to actually jump into our Q&A section. So I would like to encourage our audience to post any questions if they have in the chat available in this Zoom meeting. And I have a couple of that came in. So let me start with the first that we have on the list. And please, as you have more questions, feel free to type them in. So first question that we have is about the hook effect that could be observed on ERAP1 and 2 melt curves in the case of the compound treatment. Can you please elaborate on that?
Yes, so I think this was something that threw us off at the start. And it's essential to keep in mind, it's like a molecular crowding effect. So when you first start at the lower temperatures before any of the targets have started to be denatured by the heat challenge, it's everything in that lysate mix. And as the temperature increases, you're starting actually to enrich the soluble fraction of your lysate for your target that's stabilized. You do then see that bump, and it's all about the access for your antibody to the epitope. So as actually your lysate essentially kind of purifies out and enriches for your target, that's when you do see that enhanced response to your target. So I think when we first started doing it, we spent quite a lot of time at the start because we thought maybe there was something we were doing wrong, but actually, and you'll see it on where you see good compound stabilization, this becomes more and more exaggerated. For example, for the ERAP1 HAP10, where we saw very minimal stabilization, there's almost no evidence of the hook effect whatsoever, as compared to where we saw good stabilization, and it's really quite exaggerated.
Yeah, all right. Thank you for explaining that. We have a couple of more questions. Another one would be about the general criteria for target to be feasible for CETSA. So what would be general consideration for the target to be possible to run through CETSA assay?
Yeah, okay, so I think first off, you want a good quality antibody. You need to be getting a good response. So you're sort of thinking about what's the expression level for your target in your cell line of interest. You need, if you're going to want to measure EC50s, you're going to need a decent assay window sort of between your compound stabilized and your denatured. So you sort of need enough that you can get enough points on that curve to be sort of generating sensible EC50 fits. A tall compound at the start is good as well. I think you could do it without, and I guess just start screening, but that's at significant risk. So I'd say a decent tall compound at the beginning, and sort of being in the region of the potencies you want to work with. So like we'd had to make that adjustment after the first stage of the ERAP1 assay development, as we realized actually we were quickly going to be dropping down below that potency range. We're sort of going to need to shift where the assay sensitivity was.
Yeah, that sounds good. And we have still quite some time and a couple of more questions coming in. So the next one is, how did you handle the effect of heat shock temperature on EC50 across different ERAPs when making decisions around SAR?
Did you want me to take that one, Juliet? Or
yeah, if you'd like to.
So, I mean, I think that kind of comes back to what we said at the start for what we were using this assay for. And there were, several reasons. One of them was to build the relationship between what we were seeing enzymatically and then what we were seeing in cell function systems. And this is clearly a stepping stone between the two. And the other is, given what Juliet explained in terms of the polymorphic nature of the enzyme, CETSA kind of creates a relatively unique environment in which we can look at semi-quantitatively the engagement of molecule with the different haplotypes of ERAP1. But when it came to making decisions on SAR, we didn't really center it on CETSA so much. We use CETSA more as a kind of as that kind of confirmatory confidence builder in our screening cascade. So the enzymatic was our primary driver, enzymatic potency, and then cell function was our kind of next driver. But we built up the relationship in the same cell line that we had CETSA, which was CHA, and then the cell function readout as well. So we try to join everything up, obviously, in HAP2 all the way, as it were. So, Juliet, do you have anything to add from your perspective on the SAR decision making?
Yeah, that sounds good. Thanks, Pete. We have another questions coming in, and the next one is: have you considered to combine the CETSA experiment with a higher throughput detection system, such as TR-FRET, to test more compounds, or there are limitations? So
I think, Pete, maybe correct me here, I'm not sure how developed the FRET setup for CETSA was when we were doing this work originally. And I think because we weren't really sure what we were going to see when we started out, sort of working with all the different haplotypes, sort of the risk needed to sort of develop the additional reagents for the FRET. I think wasn't really something we considered at the time. For what we needed to do, the navigate method was sufficient. It was probably on about the borderline of how many compounds you'd want to put through that method. And I think because we were sort of confirming engagement rather than sort of using it as the primary, it was sort of the secondary level, that level of throughput was sufficient. But definitely, I think if you wanted to go much higher, you'd want to really look into FRET systems.
Yes, and maybe a short comment on that from my side is that we also have a CETSA Navigate HT format that is for more high throughput screens, which is based on alpha lysa detection chemistry and dual antibody detection. So this is a format of CETSA more adjusted for 3 to 4 well plates and screening of larger libraries up to 500,000 compounds, for example. So yeah, that would be probably way to go if somebody is interested in really high throughut CETSA based screen. Yes, we have also a question starting with compliments about the great talk. So thank you about that. And given the large EC50 change seen comparing melting at 47 degrees to 49 degrees, about 5-fold, do you think it's possible to extrapolate down to 37 degrees or perform an EC50 versus temperature at more temperatures and use Van Hoff-like analysis?
So I think, I remember we sort of briefly looked at going the other way. I think in an odd way, you might be muddying the waters if you go down, kind of coming back to that hook effect in that you're working in lysates that have pretty much everything there. And I don't know if that then you might be in a situation where you're too often you're masking access the epitope of your target if it's not sort of, you've not implemented your heat challenge and started the denaturation. That's kind of my best guess at that. I don't know if you can add anything, Pete.
Not on the technical side. I think, yeah, there was both the technical component, which you kind of talked to there, Juliet, and then there's the kind of pragmatist piece, which was coming back to my earlier point. We felt we could generate pretty accurate EC50s through our other assay systems. And so what we wanted from this system was an ability to look across the haplotypes, but also make sure that there was within reason parity in the EC50s we were generating in the CETSA system. So I
think it was a case of keeping in mind, as long as we knew that those EC50s, there was going to be a difference, that we weren't taking them as indirectly comparable, that was sort of sufficient for what we needed to use them for.
Yeah. Okay.
I think the second part of your question was similar to what someone internally who's more familiar with sort of receptor pharmacology had suggested a setup for that. But then I think, again, we've sort of come back to the EC50s was sufficient for what we needed. But I think if you wanted to look into it to that extent, that would probably be the right way to go.
Great. We have one more comment that is impressive to generate data across so many cell lines. Did you need bespoke optimization for each cell line around target expression level, cell numbers, or Western blot conditions?
So we were quite lucky in that actually pretty much one protocol covered it all. We did work with a pretty high cell number, and I think we did it on the basis of cell number. So regardless of cell size, all of the cell lines use the same cell number for the solution when we were doing treatments. I think you could probably certainly go down on some of them. For us, it just made it easier to manage, to keep that across. That to some level, I think, compensated where we had variability in the target expression, but mostly because we've got a pretty high cell number in there that was compensated for. I think a lot of that comes back to if you didn't have as good an antibody, if you did have a much lower expression level, then you might find you'd have to sort of play around a bit more with individual cell lines. But I think we sort of compensated for the little bit of variability. I think from the slide where I was showing the ERAP2 level expression, you can see actually between the ERAP1 cell lines, there's quite a degree of variability, say from the C hard to the PC3 cells. And that was something that was looked at the start, but we were fortunate that the conditions we could find that there was one that fitted for all.
All right, thank you very much. I don't see any more questions coming in. So I think that would be it for our Q&A questions from the audience. So with that, I would like to thank again our today's speakers, Peter and Juliet. Thank you very much for coming in, for sharing the great story, great data with all of us. And I would like to thank our audience for engaging in the really nice discussion. So this webinar was also recorded and will be available to watch on demand later on our social media platform. So please stay tuned to watch it again or share with your colleagues. And definitely stay tuned. We'll be coming back with more webinar series next year. So let us all know if you're interested in particular topics. You can also go to our website, pelagobio.com, to learn more about CETSA technology and basically reach out to us if you have any further questions. So thank you again, Peter. Thank you again, Juliet, and thank again to all of our attendees.
Thanks, everyone.
Thank you. Bye.
Bye-bye.
Bye.